Today we are going to learn biological synthesis of nanomaterials. In today's lecture, we are going to learn about what is biological synthesis, what are the advantages of biological synthesis of nanomaterials, and we are also going to learn the role of bacteria, fungi, algae, and plants in the synthesis of nanomaterials. In today's lecture, we are also going to learn about viral nanotechnology. At the end of the lecture, we'll also have a practical demonstration to understand the biological synthesis of nanomaterials. Let us see what is biological synthesis. Biological synthesis is a eco-friendly and sustainable method for synthesizing the nanoparticles using biological entities such as plants, bacteria, fungi, algae or viruses. And this method utilizes the natural biomolecules. For example, we can use these enzyme proteins or polysaccharides or secondary metabolites which can act as a reducing agent, and it can also act as a stabilizing or capping agent during the synthesis of nanoparticles. When compared to the chemical synthesis, in biological synthesis, we are going to use the biological molecules. It can be enzymes or it can be proteins or it can be polysaccharides. So, that is the advantage of this biological method. And this biological synthesis has 4 important phases. The first one is the initial phase. In the initial phase, metal precursor is added to the microbes or plant extract. Then in the activation phase is comprised of metal ion reduction and nucleation mediated by the microbial enzymes that can be detected by the color change in the reaction mixture. The third one is the growth phase. In the growth phase, it leads to the amalgamation of the nucleated metals into varying size and shape nanoparticles that are thermodynamically stable. And the fourth phase, that is the termination phase. So, that decides the final morphology of the nanoparticles, which is greatly affected by the different factors including the temperature, pH, time, and what is the capping agent and also the agitation frequency. So, this is the bioreduction process for synthesizing the nanoparticle through biological method. As I told earlier, to synthesize nanoparticles through biological method, we can use these microbes or we can use the plant phytochemicals. And these phytochemicals and enzymes which act as a reducing agent as well as the capping agent. When we add the metal salt as a precursor, so that will be reduced into metal nanoparticle, and the size and shape of the nanoparticle depends on the reactant concentration, pH, kinetics as well as the mixing ratio. So, once the nanoparticle is formed, then we can purify and characterize using various characterization techniques. And the purified nanoparticle can be in the colloidal form or it can be powder form by lyophilization. So, we can do the freeze drying to make it in the powder form, and we can use it for various applications including imaging, drug delivery. Not only the microbes or phytochemicals, we can also use the food waste and we can prepare the food extract, and using that we can synthesize the nanoparticles. Let us learn about biological reduction in detail. Similar to the chemical method, here also we will be using metal salt as the precursor, and we can use the microbes or we can use the phytochemicals from the plant extract, or we can use the food waste extract, so which act as a source of biomolecules such as alkaloids, enzymes, flavonoids, proteins or polysaccharides. So, which can be useful for reducing this metal salt into metallic nanoparticles, and this will act as a reducing agent as well as it act as a stabilizing or capping agent. So, as you know from the previous lecture, the first step is nucleation followed by growth of the nanoparticles, and these nanoparticles should be stabilized with the help of capping agents or stabilizing agent. So, as I told earlier, here the enzymes or the proteins which act as a capping agent as well as a stabilizing agent, and finally what you get is the capped and stable nanoparticle. Let us learn about the advantages of biological synthesis. So, the biological synthesis is a eco-friendly and green method because it avoids the use of toxic chemicals, and again it provides the biocompatible nanoparticle because we are using the biological agents as the coating agents or stabilizing agent, and again it is a cost effective technique because we are using the inexpensive raw materials like plant extracts, and also it is easy to scalable because we are using the simple plant extract or microbes. So, we can easy to scale up to industrial production, and again we can use this biological method and synthesize variety of nanoparticles including metals and metal oxides, and we can also make nanoparticles of different shapes and sizes, and again it is an energy efficient technique because it requires mild conditions. And here the reduction and stabilization is in one step, so it is simplifying the synthesis process. And also it can provide a better functionalization because we are using the biomolecules as a functional group for capping or stabilizing. And the nanoparticle synthesis through biological method has wide applications including medicine, agriculture and environmental remediation. Let us learn about how we can use bacteria for nanoparticle synthesis. Here the bacteria can be termed as microfactories for nanoparticle synthesis because large quantities of bacteria can be easily cultured in routinely used growth mediums, and they produce various enzymes and biomolecules which can act as a reducing agent and stabilizing agent for synthesizing the nanoparticles. Here the bacteria can synthesize nanoparticles in two ways. The first one is intracellular synthesis and the second one is extracellular synthesis. In the intracellular synthesis, bacterial cell absorb the metal ions and reduce them into nanoparticles using enzymes like nitrate reductase. And in case of extracellular synthesis, bacteria secrete enzymes or metabolites into the culture medium, and these extracellular agents reduce the metal ions and stabilize the nanoparticles. Let us learn how bacteria synthesize nanoparticles through intracellular route and extracellular route. So, to synthesize nanoparticles using bacteria, so the bacteria has to be grown in a petri dish at optimum conditions. Then inoculate this bacteria into the culturing flask and incubate in the optimum conditions. So, once the bacteria is grown, then you can collect this bacteria and centrifuge it in a centrifuge tube. So, once you do centrifugation, then you get this cell pellet. So, that is the biomass. So, this biomass can be collected for synthesizing nanoparticles through intracellular route, and the supernatant can be collected for synthesizing nanoparticles through extracellular route. So, here wash this pellet with the sterile distilled water, then add the metal precursor, then incubate at optimum conditions, then collect the nanoparticles through centrifugation. So, here in the intracellular route, what happens is the nanoparticle is synthesized inside the bacteria. So, to get these nanoparticles, we have to lyse this bacterial cell wall. So, this is an additional step in the intracellular route. Whereas, in case of extracellular route, you are taking only the supernatant, then adding the metal precursor, then incubating at optimum conditions, then you can collect the nanoparticles by centrifugation. Here, there is no additional step, you can directly get the nanoparticle as you are having this reducing agent and stabilizing agent in the supernatant. So, this is the difference between the intracellular route and the extracellular route. Let us learn about the mechanism of extracellular and intracellular synthesis of nanoparticles by bacteria. So, once you add the metal precursor salt, then the metal ions will be attached to the bacteria through electrostatic interaction, and these metal ions will enter into the bacterial cell, and inside the bacteria you have the reductase enzyme, and this reductase enzyme will reduce your metal ions into metal nanoparticle, and this metal nanoparticle can be stabilized with the help of protein or the enzymes or the polysaccharides which act as a stabilizing agent or capping agent. So, this is a intracellular synthesis of metal nanoparticles. In the extracellular synthesis, the metal ions will be reduced into metal nanoparticle through membrane redox protein. And another approach is the reductase enzymes will be secreted out of the bacteria, and once these enzymes are secreted out of the bacteria, then these enzymes can reduce these metal ions into the metal nanoparticle. So, which can act as a reducing agent and also it can act as a stabilizing or capping agent. So, this is the difference between the extracellular and intracellular synthesis of nanoparticles by bacteria. So, these are some of the examples of bacteria used for nanoparticle synthesis. For example, if you are using this bacteria, we can synthesize cellular nanoparticles of size between 10 to 30 nanometer. Let us learn about biosynthesis of magnetic nanoparticles. To synthesize magnetic nanoparticles, we have to use the magnetotactic bacteria, and these magnetotactic bacteria orient and migrate along geomagnetic field lines based on intracellular magnetic structure called magnetosomes. So, these magnetosomes are membrane bound magnetite particles. For example, we can use this Magnetospirillum magneticum bacteria to synthesize the magnetite nanoparticles, and here this bacteria will absorb the ions from the environment and reduce them to form magnetite nanoparticles. The advantage of this magnetotactic bacteria is once the magnetic nanoparticle is synthesized inside the bacteria with the help of external magnetic field, we can arrange this magnetic nanoparticles in the shape whichever we want. So once the bacteria is arranged, we can add the detergent and we can lyse the bacterial cell wall, then we can get the arranged magnetic nanoparticles. So that is the advantage of this magnetostatic bacteria for synthesizing the magnetic nanoparticles. Let us learn about nanoparticle synthesis by fungi. So here the fungi act as a natural, environmentally friendly biofactories for synthesizing nanoparticles due to their ability to secrete enzymes and metabolites that facilitate the nanoparticle formation. So most of you know the fungi is a very good source for producing the proteins and enzymes. So it is a act as a eco-friendly agent for synthesizing the nanoparticles. And using the fungi, we can synthesize various nanoparticles for various applications. And similar to the bacteria, the fungi also synthesize the nanoparticles by intracellular as well as the extracellular process. And the fungal biomass has lot of proteins and polysaccharides which act as a reducing agent as well as a stabilizing agent similar to the bacteria. Whenever we synthesize nanoparticles through extracellular route, there are some challenges. For example, the growth medium also contains glucose and protein, which can also act as a reducing agent and stabilizing agent. So to overcome that, we can use the synthetic media and sucrose. But using the synthetic media reduces the number of reducing agent, but the residual glucose or hydrolyzed sucrose can still contribute to the nanoparticle formation. So if you want to understand the nanoparticle synthesis through the fungi originated components, we have to wash and process the mycelia to remove the growth media remnants. And by using the intracellular extracts derived from the cell reception, we can confirm the role of fungi originated compounds such as metabolites, enzymes in the synthesis of nanoparticles. So in this research article, so they have used a 3 approach to confirm and prove that synthesis of nanoparticles through the fungi by the extracellular fluid, autolysate and intracellular fluid. So once the fungi is grown on the agar plate, so they collected using the centrifugation. So once you've done the centrifugation, so they use the supernatant for synthesizing the gold nanoparticle. So this is a extracellular fluid method. So then the next method is autolysate. So here they wash the biomass, then resuspend in the acetate buffer with antibiotics. So this antibiotic will prevent the bacterial growth and bacterial contamination, and they kept it for 8 days. Then after 8 days they centrifuged and they taken the supernatant for synthesizing the gold nanoparticles. And third one is use of intracellular fluid. So here they taken the biomass in the acetate buffer and using the cell disruptor they disrupt the complete cell. Then by centrifugation they collected the supernatant, then they added to the gold metal salt to prepare the gold nanoparticle. So by this way they confirm the role of the fungi originated compounds in the synthesis of gold nanoparticles. And they also confirm the size of reducing and capping agent. For example, the reducing agents in the fungal extract are smaller than the 3 kilodaltons such as glucose, amino acids and cofactors, and which can act as a reducing agent, but they do not stabilize the nanoparticles. So for stabilizing the nanoparticles, larger biomolecules such as proteins are used as a stabilizing or capping agent through the van der Waals forces or bonding with the nitrogen or sulfur atoms. So in this study they have concluded that the fungal metabolites can be effectively used for green synthesis of nanoparticles, but careful processing of mycelia is necessary to distinguish the contribution of fungal compounds from those of the growth media. So that is why whenever we synthesize the nanoparticles through the biological method, we have to distinguish the difference between the synthesis of nanoparticles through the growth media and the synthesis of nanoparticles through the particular microbes. So these are some of the examples of nanoparticle synthesis by fungi. Let us learn about synthesis of nanoparticles using algae. So these algae naturally tolerate heavy metals, so it can be a very good bioagent for removing the heavy metals pollution, and also it can be useful for synthesizing the various metallic nanoparticles. And these algae assimilate low concentration of heavy metals for essential cellular process such as enzyme functioning, electron transport and nitrogen assimilation, but high concentration negatively impact their growth and morphology. So the mechanism of nanoparticle formation in the algae is this algae combat the heavy metal stress by producing metal chelating agents and reactive oxygen species, which interact with the metal nuclei to facilitate the biosynthesis of metallic nanoparticles. So, these algae synthesize nanoparticles by three ways. One is intracellular. So, once we add the aqueous metal salt, so this algal biomass will uptake this metal salt solution and it can synthesize the metal nanoparticle inside the algae using the polysaccharides or proteins, which can act as a reducing agent as well as the stabilizing agent, and you can get the stable nanoparticle. The second approach is it can synthesize the nanoparticles by the extracellular method, that is it can secrete some enzymes or proteins extracellularly and which can act as a reducing agent and can reduce the metal ions into metal nanoparticles. And the third approach is the algae has lot of pigments like chlorophylls, phycobilins and carotenoids, which can act as a reducing agent as well as the stabilizing or capping agent. With the help of sunlight, it can convert the metal ions into the metal nanoparticle. Let us learn about heavy metal removal by algae based nanoparticles. The first step is biosorption. So, here the adsorption of heavy metals on the surface of cells will be assisted by metabolic or physicochemical means. Then once these metal ions will be converted into the metal nanoparticle, so that can be accumulated in the vacuoles and cytoplasm. And third step is the biotransformation, so which will convert this metal into the non-toxic metal nanoparticles by the activity of chelating agents such as peptides, enzymes or organic acid. The nanoparticles synthesized using this algal biomass has dual benefits because it is converting the heavy metals into the nanoparticles, and these nanoparticles also has the efficiency in degrading the hazardous pollutants like dyes or nitro compounds from the aquatic environment. So, when you are using this algae based green biosynthesis, it can address the heavy metal pollution. At the same time, it is also producing the nanoparticle which can degrade the toxic metabolites in the wastewater and which will be useful for water purification application. So, these are some of the examples of nanoparticles synthesized using algae. Let us learn why do microorganisms synthesize nanoparticles. Microorganisms synthesize nanoparticles as part of their natural metabolic processes. This phenomenon termed biogenic synthesis. So, is a result of interaction between microorganisms and their surrounding environment. Let us learn some of the reasons and mechanisms behind this ability. The first one is defense mechanism. The microorganisms synthesize nanoparticles to protect themselves from the environmental stresses including toxic metal ions and oxidative stress. So, these microbes will convert the toxic metal ions into less soluble and less toxic nanoparticles so that the microorganism can survive in environments with high metal concentration. And next one is oxidative stress. The nanoparticles like silver nanoparticles exhibit antimicrobial properties that may prevent the competition from other microorganisms. So, it will not allow the other microorganism to grow in the particular environment. And next one is energy and metabolism. Most of the microorganisms reduce the metal ions during respiration or metabolism to obtain energy. And some microbes use metal ions as electron acceptors, indirectly lead to the synthesis of nanoparticle. The third one is detoxification. So, here the microbes synthesize nanoparticles to regulate or detoxify excess metal ions in their surroundings. This mechanism helps maintain ionic balance and protect cellular structures. The fourth one is survival and adaptation. The nanoparticles can enhance the surface characteristics of microbial cells, which help them to adhere to surface or resist harsh conditions. And also it provides the evolutionary advantage, that means these nanoparticles might confer a selective advantage such as creating a microenvironment unfavorable to predators or competitors. And the last one is the nanoparticle synthesis by this microbe, it may be due to the enzyme activity and secondary metabolites, because the microbes have the enzymes like nitrate reductase or superoxide dismutase, which can act as a reducing agent and which leads to the nanoparticle formation. And it also has a lot of secondary metabolites like proteins or polysaccharides, which can act as a reducing as well as a capping agent for nanoparticle synthesis. Let us learn about synthesis of nanoparticles using plant extracts. So, the synthesis of nanoparticle using plant extracts is a eco-friendly and green synthesis method because the plant extract provide a sustainable and environmentally friendly alternative for synthesizing nanoparticles. So, they act as a natural reducing and stabilizing agent and eliminating the need for harmful chemicals in the traditional chemical synthesis method. And again, this is a cost effective and efficient method because the plant mediated synthesis is relatively inexpensive because we are using the readily available plants, and the process is very simple. It requires very fewer sources and it can be scaled up for industrial application also. The third one is the bioactive compounds. The presence of bioactive compounds like flavonoids, terpenoids, alkaloids, which play a key role in the reducing the metal ions to the metal nanoparticles. And the nanoparticle synthesis using the plant extracts have wide application, especially in biomedical field. So these are some of the examples of biosynthesis of nanoparticles using plant extracts. So whenever we synthesize nanoparticles using plant extracts, it has an additional advantage. For example, if you are using the medicinal plant for synthesizing the metal nanoparticles, the metal nanoparticles like silver nanoparticles, it has a very good antibacterial properties. And when you are using the medicinal plant like neem, so which will also have antibacterial property. And when you combine these two and synthesize the nanoparticles, which will have a synergistic effect. So, for example, if silver nanoparticle can kill 5 bacteria and neem can kill 5 bacteria, when you are synthesizing nanoparticle using the neem extract, it can kill more than 20 bacteria. That is called synergistic effect. Suppose instead of 20 bacteria, if it is killing only 5 plus 5, that silver nanoparticle is killing 5 bacteria and neem is killing the 5 bacteria, and total it is able to kill only 10 bacteria. That means that is additive effect. But mostly when we use the plant extract for synthesizing the nanoparticles, it always shows a very good synergistic effect for antibacterial or anti-cancer activities. By this way, we can improve the therapeutic efficiency of nanomaterials. Let us learn some of the important parameters for synthesis of metal nanoparticles through biological method. Whenever we synthesize metal nanoparticles through biological method, we have to optimize the conditions. For example, the size and shape of your nanomaterials, everything depends on these pH, agitation, temperature and the metal salt precursor concentration and the ratio of reactants and the incubation period. By optimizing these parameters, we can successfully synthesize the nanoparticles through biological method. Let us learn about viral nanotechnology, how we can use this virus for making the nanomaterials. Viral capsids devoid of their nucleic acid genomes can be thought of as nano containers. That means when you have the virus and this viral capsid is made up of protein, and most of you know the size of virus are in the range of nanometer. And when you remove this genomic material, it can be DNA or it can be RNA. So once you remove this nucleic acid, we can use this protein as a nano container, and once you remove the nucleic acid, we can add anti-cancer drug and we can use it for cancer therapy. So this is called as viral like particles or viral nanoparticles, and this is inspired by nature, that is a biomimetic approach. And we can also use this virus capsid to serve as a size constrained reaction vessel. For example, you can see here, so the metal ions can enter into the viral capsid, and if the size of the virus is in the range of 20 nanometer, and the metal ions can enter into the viral capsid through diffusion. So once it enter, it will be reduced into metal nanoparticle and it will grow. The maximum growth it can attain is it can reach up to 18 or 19 nanometer. So it will have a uniform size nanoparticle because the virus capsid act as a kind of size constrained reaction vessel. So we can easily synthesize uniform size nanoparticle. If you want to make uniform size nanoparticle, we can use the virus as a kind of nano container to synthesize the nanoparticles. And once the nanoparticle is synthesized, we can remove this viral capsid by treating with a detergent or by using the protease enzyme. We can remove the viral capsid and what you get is the intact nanoparticles. So that is the advantage of this using these virus for making the nanomaterials. Let us learn about viral nanoparticles functionalization. In the viral nanoparticle, once you remove the nucleic acid, we can add the anti-cancer drug or any therapeutic molecules inside the virus, and in this subunit interface we can add the gadolinium metal, and which can be useful for imaging, which will be useful for the cancer diagnostics. And to make this nanoparticle specifically to the cancer cells, we can add the RGD. So this is a 3 amino acid peptide, arginine, glycine and aspartate, and most of the cancer cells overexpress the receptor for the RGD. So this can easily go and bind specifically to the cancer cell. So by this way we can make the multifunctional nanoparticle. Inside we have the anti-cancer drug and we have the gadolinium metal, which can be useful for cancer diagnostics by imaging, and the outside exterior we have the RGD, and which will specifically bind to the cancer cell. So in this way we can target these nanoparticles specifically to the cancer cell, and we can increase the therapeutic efficiency and reduce the side effects. So the next question is, what kind of virus we have to use it for synthesizing the nanomaterials? We have to use the plant virus. Why we have to use the plant virus? Because to minimize the chance of virus interacting with the human proteins and causing toxic side effects, infection and immune response. To avoid that, we have to use the plant viruses. And additionally, the plant viruses are easy to produce in large quantities, and plant viruses are available in various sizes and shapes. So here also you can see here it is available in various sizes and various shapes. So that is the advantage of the plant viruses. And again, the plant viruses can hold approximately 10 cubic nanometers of particles. So it can have very good drug loading efficiency. So it can carry many molecules of cancer drugs, and it can be very useful for cancer therapy. So that is the advantage of using this plant virus. Additionally, when we are using the rod shaped plant virus such as tobacco mosaic virus, the amino acid composition of this tobacco mosaic virus is different for exterior and interior. So in this case we can synthesize different kind of nanoparticles inside the TMV and outside the TME. For example, we can synthesize the silver nanoparticle inside the TME virus, and we can synthesize the gold nanoparticle or some other material on the exterior of the TME virus. So in this case, this can act as a template for nano rod synthesis. By this way, we can make a novel and new materials. Let us learn how to produce viral nanoparticles for various therapeutic application. So to produce the viral nanoparticles, we have to add the virus to the plant leaf, and once we add the virus, it will make millions of copies of plant virus. Then these viruses can be chemically modified. We can remove the nucleic acid and we can add the anti-cancer drug or any therapeutic molecules inside the viral capsid. Then we can also add antibody or the ligand which can specifically bind to the cancer cell. Once the viral nanoparticle is ready, then you can evaluate the efficiency of viral nanoparticle in vitro as well as the in vivo condition. For example, we can use the cancer cells which can expressing the particular receptor, and when we add the particular viral nanoparticle, it can bind to the cancer cell and it can kill the cancer cell. Then in the in vivo condition, we can have a mouse model which is having the tumor. Then when we inject this nanoparticle, it can go and bind to the tumor cells and it can destroy the tumor cells. So by this way we can evaluate the therapeutic efficiency of viral nanoparticles both in vitro as well as the in vivo condition. I hope you got the overall idea about biological synthesis of nanomaterials. Let us go to the lab and learn how to synthesize nanomaterials through biological method. Today we will learn how to synthesize silver nanoparticles using neem extract. To proceed with the experiment, we need freshly plucked neem leaves, silver nitrate, glassware, tissue paper, spatula, scissors, micropipette and microtips. In the first step, we will prepare the neem extract. We have freshly plucked leaves here. that have been washed with distilled water and chopped into smaller pieces. This is approximately 40 gram of neem leaves, which we will add to 200 ml of distilled water. Cover this with aluminum foil to prevent evaporation while boiling. Place it on the heating mantle and let it boil for around 15 to 20 minutes. Here you can see that the color of the water has changed. Turn off the heater and set the beaker aside to cool. As the solution cools, you can observe the color change due to the neem extract. Once cooled, we will pour the solution into a separate bottle. Now we will filter the neem extract. We have already prepared the neem extract solution by filtering it with Wattmann filter paper. To ensure further purification, we will filter the solution using a syringe filter membrane with 0.22 micrometer pore size. Now we have prepared the neem extract. Let us proceed with the synthesis of silver nanoparticles. For synthesizing silver nanoparticles, we need neem extract, ultra pure water, 2 conical flasks with beads, one for the blank and one for synthesizing silver nanoparticles, 10 millimolar silver nitrate solution, micropipette, microtips and measuring cylinder. First, we will add 50 ml of ultra pure water to the blank, that is a negative control conical flask. Set the magnetic stirrer to 40 degrees Celsius and 300 RPM. Next, add 50 ml of 10 millimolar silver nitrate solution to the second conical flask. Cover the flask with aluminum foil to prevent the photoreduction of silver nitrate. Set the same temperature and RPM on the stirrer. Leave both flasks for a while. Now we will add 5 ml of neem extract to both flasks. Allow the reaction to continue for at least an hour to ensure the complete reduction of silver nitrate ions. As you can see, there is a slight color change indicating that the reduction process has begun. The further color change signifies the formation of silver nanoparticles. We will leave this solution to complete the reduction process. Once the reaction is complete, turn off both the stirrers. As you can observe, the color of the solution in the blank flask containing ultra pure water and neem extract is different from the one in the flask containing 10 millimolar silver nitrate solution and neem extract, indicating the formation of silver nanoparticles. To confirm the formation of silver nanoparticles, we will use UV visible spectrophotometer. First, turn on the UV visible spectrophotometer and allow it to warm up. Open the software on the system and select the new file option. Choose spectral scan and set the wavelength range from 350 to 800 nanometer. We will first calibrate the baseline using ultra pure water as the reference. When adding samples, ensure you hold the cuvette at an angle to avoid bubble formation. Place the cuvette in the machine, ensuring the clear sides align with the light source and detector. Select the blank calibration option. After calibration, perform a blank reading with ultra pure water, which is the solvent we used. And press the 0 option on the software. Once completed, discard the water and add the sample to the cuvette. First, we will take the UV reading of the 10 millimolar silver nitrate solution. Press start. As expected, there is no peak for the silver nitrate solution. Now wash the cuvette and add the neem extract. Place the cuvette inside the machine and press start. Again, no peak is observed in the range of 350 to 800 nanometer. Next, we will add the synthesized silver nanoparticles. Since it is highly concentrated, we will prepare a 1/4 dilution of the stock solution. To do this, we will mix 250 microliter of nanoparticle solution with 750 microliter of ultra pure water to achieve the desired dilution. Mix well and transfer the solution to the cuvette. Place it in the spectrophotometer and take the reading. we can now observe a peak around 450 nanometer, confirming the synthesis of solar nanoparticles. Save the data, finally watch the cuvette. This concludes our demonstration on the synthesis of solar nanoparticles through biological method using neem extract. As a summary, in today's lecture, we learnt about biological synthesis of nanoparticles and advantages of biological synthesis. And also, through practical demonstration, we learned how to synthesize nanoparticles by biological method. Thank you for your kind attention. I will see you in another interesting lecture.